Progress.
Hello.
In.
I have a question, Professor.
What was that? I'm sorry.
I have a question.
What's that?
I'm not doing the extra credit practice questions.
Okay.
So I want to know if it's not going to affect my,
It won't. If you look, and I don't know if you see it or not on your end, but the only way that it actually eventually affects your grade is if I put in zeros for when people don't do it.
Okay.
And I'm not doing that.
Okay.
It will improve your grade, but it won't hurt your grade either. It's extra credit.
Okay. Thank you.
See if we get some more people. We don't have very many people here. I wonder if it's the weather.
Just so you know, I'm here, but my computer is
Right.
finally, I'm still here having
Okay.
them fixed here.
I understand.
Thank
Where are
you.
you at?
At Best Buy, I have a membership with them, and it's the greatest thing ever. Like, they fix anything and every issue I have had with like
Okay.
my cars or anything tech. So it's been like really great experience having a membership with them. So they're fixing my computer right now as we speak.
Okay, I understand. Let's wait just one more minute and see. We just don't have a lot of people here tonight. Let's see if we get some more people. All right, I'm gonna wait just one more minute until like 6:01 or 6:02, and then we'll jump into it. Just want to see if we can get a few more people here. Let's just see. So I realized that I gave you the wrong due date for the lab exercises. I think I put on there that they would be due today when they should have been due last night, because that's the way they've always been. But because I did that, the lab exercises that we did last week, lab exercises 16 and 17, they won't be due until the 25th. And so that's Friday. So if you did not get them done, realize they're still open for you.
I'm sorry, did you say the lab exercise?
Yep, the lab exercises 16 and 17, which we was the nervous tissue in the spinal cord, which we worked on last Wednesday.
Okay, thanks.
Okay, I think let's get into this now. We've got more people than we had before. So like I said the other night, I would prefer to not do a lab rather than get more behind in lecture. And so what I'm going to do is, if you look at the syllabus, there's an autonomic nervous system. I'm going to just not do that lab. We haven't done it in past semesters. I don't think it's going to hurt to not do it this semester. I haven't done it in past semesters. And so what I'll do is I will give it an assignment. I'm going to give it a title. I'm going to put it in the lab book, but I'm just going to give everybody 10 points for it. And so when you see that, you'll know why that's there, is that I'm just not gonna just not do it, because I would prefer to work through the nervous system and get through part of the spinal cord tonight, and then do the brain in lab next week. Any questions or concerns?
Did I miss anything?
I don't know. I can't tell you that. The only thing I can tell you is that I think I said that the due date for the lab exercises was the 23rd, which would be tonight for 16 and 17. They should have been due last night, but because I didn't make them due until the 23rd, I'm giving you until the 25th to do them. So if you haven't done them, please get them done. I think when I looked at it earlier today, about half of the class had not done them. So please get them done. And let me jump into lecture. That's the only thing, Lauren, that I can think that you missed.
Professor,
Okay, just making sure.
will you be sending an email after tonight of what will be due moving forward, since we're doing like lecture instead of lab tonight?
It's almost the same thing.
Okay.
Let me see.
I wasn't sure if there was going to
For a
be
second
anything
here,
in addition.
because I didn't put any due dates, different due dates. Bear with me just a sec. So this is the email that I just sent you the other night.
Yeah, I did like all of the chapter 7 except for the smart book because it was closed. The date was closed before you extended it on Sunday. And then the lab. So I just wasn't
You
sure.
should be able to do it now.
Yes, I went back last night and
Okay.
got in it. So I'm fine. I just wasn't sure if you were going to send an email, if there was going to be anything extra due besides what you sent.
If I do, I'm not going to send it. I won't have time tonight. There's no way for me to do it tonight. I forgot about doing something that I should have done 3 weeks ago for the chair of HAC. And so that's what I've been working on for the last couple hours.
Okay, yeah, that's fine. That wasn't an expectation of it being tonight. I just wasn't sure, since we were on track for lab. So if we did a lab tonight, then obviously something new would be due. I just wasn't sure if the same assignments were due, like the same dates, or if there was going to be an addition from tonight.
Let me, bear with me here. I don't, because like honestly, I send out those emails for each of my classes, and I have 6 classes. I can't remember sometimes what the due dates are in this class versus one of my other classes. So let me, and where we are, unless I make a note of it, which I make a note of where we are in lecture, but that's it. Bear with me. Okay. So what it says that, well, no, wait, this was for before. Darn it, just a sec. I know I had more than that.
If it helps, I don't know if you sent them all out on the same day, but you did send it on
I
Sunday.
did.
Okay.
March 20th. Maybe it's on here. Okay, just a sec. Oh, I'm gonna just let you know if there's any changes in due dates. Give me 1 second. I have to look and see what the due dates were first.
Will it help if I tell you what it says? I do have the
Actually,
email.
I can find it. Give me 1
Okay.
second. I've
Okay.
almost found it because I
I
found
just didn't want to make it an inconvenience for you.
I
All right.
appreciate it. I appreciate it. And I think this is gonna bring up the wrong thing, but let's see here. That's it. One second here. This class. Okay, so what it says is that the lab exercises 16 and 17 are due tonight. The virtual lab is due tonight. The bones case study is due tonight. If you haven't done it yet, just submit it. The Dropbox is not going to close. It's set up to stay permanently open. Just submit it. The Smart Book Chapter 7 is due on the 27th. The Smart Book quiz is due on the 27th. The Chapter 12 Smart Book and quiz will be due on April 4th. None of these deadlines are changing.
Sounds good. I just wanted to make sure.
Thank you. I
Thank you.
appreciate you doing it.
Professor Gessel, sorry, I had a little hand up for a while. I did that, I guess it was lab exercise 17, like yesterday, and a lot of the questions had a real picture.
On a what?
Had a real picture of like a section of, you know, like the spinal cord.
Right.
We identify things. I could never find something similar. I could only find, you know, like the drawn diagrams, and I had a really hard time identifying some of the parts. I mean,
Christina, weren't those the figures that I modified for you that I gave you the answers?
I don't remember that. I had the same problem.
I don't know, but I basically had to use the correct answer for almost all of them.
Bear with me just for a second.
Yeah, I did too.
Because I thought those were the ones that I gave to you. And I actually pulled the answers. Bear with me just for a second.
I mean, I'm not as worried about, you know, since we had unlimited attempts, I just want to be able to understand what I was looking at, you know, so I can identify it on my own in the future.
Give me 1 second here.
Yeah, I think I even struggled with that one. I had to use the book a lot or guess in a way. And a lot of it was like pictures of like the like pia mater, where the ganglion root was, and the
Oh, you know what?
posterior
The
like
anatomy,
that.
the anatomy.
Yeah, a
Was
lot of it was like
bear,
that.
bear with me. Wait 1 second. I'm sorry. Let me get to the questions because that's what I'm doing right now. Give me 1 second here. Right here. Do you remember specifically what question number it was?
Wait.
There were several. I feel like maybe one was like 9 or 10.
Okay, so 9 or 10.
I think.
I got 6 out of 43 and 7 and 8. Wait, no, 7. 8.
Okay, maybe try 8. It was somewhere around there.
Yeah, it was 6, 7, and 8. They were 5, 6, 7, and 8 actually.
Yeah, it was all the spinal cord nerves.
Yes.
Let me give it 1 moment. It just has to open up. It's a lot of information, so it takes it a minute to open up. Hopefully it will open up. Maybe.
I can text you a screenshot if you'd like to see.
You know what? It'll probably be faster for me to just try to pull it up here. Give me 1 second. Maybe it will be faster. There. So you said question 8 was one of them?
It was one of them. Yeah, it was like from 5 to 8.
Okay.
They were all the same picture, just different. Yes, there you go.
Okay, so the posterior ramus. If you, let's see here. Actually that right there. It's that right there. But I'm going to see if I can't, bear with me here just for a second here. So it's that right there. And it's that on this side. The anterior ramus is right here. Posterior ramus is right here. If you look at this one, it's moving back towards the back. Posterior meaning towards the back. It's that one right there. Does that help you at all?
Little bit. I think the structure doesn't say if it's posterior or anterior in the question, so
Let
it
me
just
look
made
at
it
it.
a little
I'm
harder.
come back and look at it again. Give me 1 second here. No, and it probably doesn't. I'll see if I can show you how to distinguish what you from back here. Give me 1 second here. Hazers. Sorry, because I switched pages, it has to reload it. So give it a second. Maybe. Second longer. So if I'm looking at Louis, really? Sorry, you guys. If you look right here, bear with me, see. This is the anterior side. This is the posterior side. This right here is the spinous process. If you look at a cross section through the spinal cord, you'll see that this is the anterior part. This is what the anterior horn looks like. Posterior horn looks like this. Therefore, this is going posteriorly. Does that help? A little bit, maybe.
Yeah.
The more you look at these things, the easier they get. But welcome to looking at cadavers. The longer and the more, the reason I put these in here is to give you exposure, because if you're probably learning really quickly, and I hope you gain an appreciation of it, that what you see on a diagram looks very different from what you see in a cadaver. And a lot of schools use only models and diagrams, and you don't use cadavers, which is not really helpful sometimes.
I think because it's so up close that you can't see the actual position that you're looking at, like whether it's straight on or if it's turned this way or if it's turned that way. So that's why for me, I was like, okay, so posterior, anterior, like my head, I was trying, looking at the other pictures that weren't cadavers, associate different parts that I could try to, and what helped me was the two, the holes, what are they called?
Two holes.
The small holes here, that are
These right
no.
here, or these?
The two bigger holes, and then the small hole beside it. What are those? Bring the arrow down, on over here, no, to the right, or no, the open holes, there you go.
Okay.
They kind of helped me decide what position it
Oh,
was
so
in.
what were you looking at there, Miss Sherry?
I don't know. It was, I actually, when I first pulled this first picture up, I sent a text to Justin with the picture and I said, what is this? I said, I have not a clue.
You want me to tell you what you're looking at?
Please, yes, yes.
I think because this is bifurcated back here. You're
Okay.
looking for a cervical vertebrae.
Okay.
And the only reason I know that is because this is bifurcated back here. It's got a split. This, I think, is the jugular vein. This is the carotid artery.
Oh, okay. Okay.
And the reason I know that's a vein is because you can see which one is less circular in shape.
All right.
They're both vessels, and you can see that, but arteries maintain their shape better than veins do.
All right.
This right here, this little tiny guy right, oh, is it right there? No, would it be right there? No, it wouldn't be right there. Never mind. I was going to say something, but I think I'm going to be wrong, so I'm not going to say it. Wait, would I be right? Bear with me really quick. I'm going to look at a cervical vertebrae because I think that's a transverse foramen, and I'm not sure. Images. I want to see just one. So this little hole right here is a transverse foramen of a cervical vertebrae. You see the bifurcation. This is the C2 vertebrae. And so when I'm looking at this, that is right. That right there, this right here, and maybe over here somewhere would be the transverse foramen. But I can tell you right now, just looking at this, and I'm gonna show you something else. Spinal cord. So we're looking at this, and this isn't labeled, but this is that same thing. That's not a cadaver picture. This is the posterior horn. So this is the posterior side where the spinous process is. That's the posterior side. So looking at this right here, yeah, you're looking at it from the side, but
Right.
This is the posterior horn. This is the anterior horn. This is the spinous process.
Okay.
This is a cross section through the neck. Does that help a little bit?
Yeah, it does.
Okay.
It does.
I think right here, this little area right here, I think this is about where the trachea would be, or slash thyroid cartilage. So this right here. And your esophagus is back here. Because if you think about it, your carotid artery is right posterior, posterior and lateral to your trachea. And your esophagus is kind of smashed up behind your trachea. Does that help at all, guys?
Mhm.
Let me see what else we've got here. So this was probably not a hard one for you.
That was easy. So it wasn't that one.
Okay, so this one, anterior ramus, and the way I would orient myself with regards to this right here, because this is the anterior, this is the posterior. Anterior, posterior.
Mhm.
What's the difference between like when it's talking about the roots, like if it's posterior root versus posterior ganglion? Like they're so close to each other.
The ganglion is this. Remember what a ganglion is. In the peripheral nervous system, which is anything outside of the spinal cord and the brain, the ganglion is a cluster of cell bodies. It's a bulge. This right here, meaning that this bulge right here, this is the posterior root ganglion or the dorsal root ganglion. Remember that dorsal and posterior are the same thing. These are the posterior roots. These are the anterior roots. This bulge right here is the ganglion. This is the posterior rami. This is the anterior rami. Does that help a little bit? Okay.
Yeah, thank you.
Of course. Let anterior root. Yeah, you can see this just doesn't do it justice. But there's your dorsal root ganglion. Here's the dorsal or the posterior root. Here's the anterior root. It just doesn't do it justice because you're looking at a model versus a cadaver. Right there is your posterior rami.
I feel like this one was really hard to see some of those, like 1 and 2, like just I mean, they're just so close together, like the little tiny, structures.
I can understand that.
I was like trying to zoom in on the PowerPoint.
What?
See, I was trying to zoom in on the PowerPoint diagram that was very similar so I can figure it out.
Did it help to zoom in on it?
A little, but it only got so big. It didn't get as big as I wanted it to.
Okay. I tried to enlarge them. I tried to actually enlarge them.
I got a little tripped up at some of the questions at the end. It was like asking like if a sensory synapse or something sent a message or something to the anterior spinal cord or whatever, where would it send the rest of the message to like, and then
Bear with me. Do you remember what question it was?
it was somewhere between 30 something and 40. It was asked quite a bit of questions like that where.
Okay, bear with me. Let me look at it. Give me 1 second.
They started on question 38. There's 38, 39.
So where is the?
Yeah, there's one of them.
Let me see here. Where is the posterior dorsal root ganglion located in sequence of structures through which sensory impulses pass as they enter the spinal cord? It's between the spinal nerve and the spinal root. Where's the spinal nerve and the spinal root? If we're looking at this picture right here, this is the spinal nerve. This is the spinal root. Therefore, the ganglion is right there. I'm gonna flip to another picture. So here's the root, here's the ganglion, here's the spinal nerve. So it's right there. Where is the posterior root located in sequence of structures through which sensory impulses are going to pass as they enter the spinal cord and travel towards the brain? Between the spinal root and the gray horn. So this is the gray horn right here. Here's the posterior gray horn. This posterior root's right here. Where is the posterior gray horn located in sequence of structures through which the sensory impulses pass as they enter through the spinal cord and travel toward the brain? It's right there. So it's in between the posterior root and the white column. Do you remember what the white columns are? They're the tracks that are going to go up towards the brain.
I think maybe I was just reading it wrong. Like I didn't realize it was asking like, where's the posterior gray horn? I think I was just the way it's worded. It was tripping me up maybe.
I can understand that. Okay, other questions that I can maybe help with? And I put the word maybe in there really because sometimes I'm not very helpful. All right, let's jump into tonight. I want to finish this tonight. I want to get started on the spinal cord. You'll see that the spinal cord and the brain are going to go a lot faster. So let's finish up chapter 12 and move into chapter 13. And next week, I want to do the brain in lab. And like I said, I have a sheep brain that I want to dissect. There's really not a lot to it because it's so small, but you really can't get them otherwise. But I did get from this butcher that I've been working with, I did get some sheep brain that wasn't fixed. And it doesn't get what the fixed brain versus the real brain doesn't, the fixed stuff doesn't do it justice because fixed brain is very stiff. The brain itself actually has the texture of like cold oatmeal. And you know that cold oatmeal is kind of like Jell-O. you'll see the same thing, that this is not really, it's very interesting in comparison. So I want to back up just a little bit, because like Justin made the comment before, action potentials, I think, are some of the hardest parts, but like Justin's Alan said, they're critical. And so I'm going to back up just a little bit to the start of this action potential. So we get the summation of these local potentials that are moving through the dendrites and the soma. And they gradually add or cancel out each other as they come in. So you've got multiple incoming signals coming into this cell body. And they are going to either add or they're going to cancel one another out. And with regards to the change in voltage of the membrane, bear with me just for a second. I'm going to see if I can switch. So I have this soma with my 5th grade looking soma, or kindergarten looking soma. With 5th graders are better drawers than that. So here's my 5th grade looking soma and their axon. And here's my dendrites, here's my soma. At multiple places attached to the soma and the dendrites, and multiple axon terminals synapsing onto the dendrites of this neuron. Some of them are bringing in inhibitory signals, some of them are bringing in excitatory signals. The excitatory signals are going to cause depolarization of the membrane. Inhibitory signals are going to cause hyperpolarization of the membrane or repolarization of the membrane. If enough excitatory signals come in, if enough of them come in, that when the signal finally reaches the axon hillock right here, the trigger zone, if the signal reaches the trigger zone that says that the membrane is above, or at least or above -55, I'll fire an action potential. But I have to reach the threshold of -55 or nothing happens. This will make more sense as we go forward, but understand that I've got to reach a threshold or nothing's going to happen at all. Once the action potential happens, it's an all or nothing thing. It's going to go to maximum and it's going to go all the way down the axon. So when a steadily rising local potential arrives as a local current at that axon hillock, and it depolarizes the membrane. If this local potential rises to the critical depolarization voltage, the threshold of negative 95 millivolts, it will open sodium voltage gated channels. It's also going to open, slowly opening potassium voltage gated channels, but they open a lot slower. But the sodium channels are going to open very fast. So we're right here. It's reached the threshold. And we start and once we reach the threshold, we get some voltage gated sodium channels opening. If at that threshold, the neuron will then fire an action potential if that threshold is reached. And the voltage gated channels start to quickly open while voltage sodium gated channels slowly start to open. A few sodium channels open, but as the sodium channels open and the sodium rushes into the neuron, the voltage starts changing and more of the membrane depolarizes and reaches that threshold of -55. And the result is that more voltage gated sodium channels will open. So it's a positive feedback loop where you reach that voltage and then some sodium channels open and they result in depolarization, which causes more channels to open. And that's why this shoots up. As the potential rises past 0 millivolts and peaks at +35, the sodium channels then get inactivated at +35. So they close. And at this point, the potassium channels that started to open down here are completely open.
Hi. Yeah. Did you have a good meal?
So they're inactivated and sodium channels are inactivated, they're closing, and the potassium channels are now completely open. So the potassium moves out into the extracellular fluid, which repolarizes the membrane, making it more negative again inside the cell. Because remember, potassium is positively charged, and as it leaves the cell, the result is that the inside of the cell is going to become more negative again. Sodium entered, making it very positive inside. But then those channels close and the potassium channels are completely open, and they make that inside of the cell, when they rush out, make the inside of the cell very positive, negative again. And so the action potential as a result has this quick up-down motion where the voltage reaches the threshold and then the voltage is going to return to the resting membrane potential. The potassium channels don't close as quickly as the sodium channels do. So slightly more potassium leaves than sodium enters, resulting in a hyperpolarization of the membrane where it becomes a little bit more negative than the resting membrane potential. That's hyperpolarization. And it's because those potassium channels are slower to close. And so they stay open for a little longer than they would. It's going to be important in a moment. Is this clear how this works? During an action potential, so as a result, during an action potential, sodium and potassium switch places. During the hyperpolarization, this time right here, sodium gradually diffuses into the cell and it returns back to the resting membrane potential of -70. Questions? So it's what it is it's a major interplay between sodium and potassium. Sodium rushing into the cell, then potassium rushes out. So looking at this diagram, and I'm going to zoom in just a little bit. Actually, you know what, I'm going to do this and then it's going to screw it all up, but it's going to make it bigger. Okay. So at the resting membrane potential, this is just showing the opening and closing of the sodium and potassium gates and their status. At the resting membrane potential, both of them are closed. Both the sodium channels and the potassium channels are closed. During depolarization, the sodium channels begin to open. Excuse me, the sodium channels are opening and the potassium channels begin to open. But because the sodium channels open very fast, the depolarization is very quick. During repolarization, those sodium channels close. And now the potassium channels are fully open and it leaves the cell. They're slow to close, the potassium channels, and as a result, we get this hyperpolarization until they close completely. So I'm going to ask some questions here. Now we've gone through that slide. At threshold, actually, what, by definition, what is threshold?
Like when you get to the point where all the conditions are right to open up the gate.
Which gates? Say it one more time, Christina.
Well, I mean, you meet the threshold that doesn't it open up the sodium gate first?
It
The
is
sodium comes out, which then makes the potassium come in, correct?
right. It's going to be where the sodium and at that moment, right before threshold, both of the gates are closed. The sodium channels are starting to open. They're being quick to open, and potassiums are also starting to open, but they're much slower. If it helps you to think about it for right now, think of the idea that the potassium gates are closed here and that it's just sodium channels and they open. What's depolarization?
It's when the sodium pump opens and the sodium comes rushing into the cell.
Yep. What's going on with regards to the separation and charge?
It becomes more positive inside.
Right. So it's actually, if you think of it this way, so what does it mean to be polar? It means that there's two very distinct separation of charge, right? Very distinct separation of charge. Depolarization is where you're making them so they're not so distinct anymore. Repolarization is you're making them distinct again, where they're starting to blend together versus they had different charges. That's where the words are coming from. Is depolarization meaning you're getting rid of the polarization? Repolarization meaning you're going back. So with depolarization, Miss Samantha, do you like Sam or Samantha?
Sam.
No. I thought so. When I responded to your email earlier, I thought I can't remember what she prefers to be called. During depolarization, which channels how are the channels? What are they doing at that moment?
Well, the sodium channels are more open versus, and then the potassium channels are slightly open.
Good job.
I believe.
Yep. Now someone else, what does repolarization mean?
The gates are starting to close and it's becoming more negative. The membrane is becoming more negative.
Which gates are closing, Justin?
The sodium gates are closing faster than the potassium.
And at this point, are the potassium gates, they're fully open, right?
Yes.
Okay. What's hyperpolarization?
Isn't that when the cell's getting back to normal and the like resting membrane potentials like back to normal? And
is it completely back to normal, or is it more negative than normal?
It's still more negative, I believe.
It's more negative than normal. That's important in a second here. Understand that, that's why I want you to think of depolarization. So when something is polar, it means that it's got, bear with me here, it's got a positive and a negative side. Inside of the cell is much more negative than the outside. When it depolarizes, they're mixing up. There's no longer that major separation in charge. When it repolarizes, it comes back. When it hyperpolarizes, rather than having this being 0 and this being minus 70, this is now minus 72 instead. So it's even more negative than it was before.
Is there a reason for that action, or is that just what happens?
The reason it happens is because those potassium gates are slow to close. And it's going to be really important in a second. So, look, overview of this. Action potentials are all or nothing. Graded potentials, they're graded, so they have different amplitudes. Action potentials, if a stimulus depolarizes the membrane enough to reach threshold, an active potential is fired at its maximum voltage of positive 35 millivolts. If it's not reached, no action potential will be generated. A stronger stimulus that possibly goes a little over threshold is not going to make a difference in the amplitude of the action potential. It's still going to go to 35 every time. If you reach threshold, you go to 35 every time. They don't get weaker with distance. Action potentials, they don't drop off with distance like graded local potentials do. An action potential at the end of the axon is not any weaker than it was at the next to the cell body. So an action potential, when it gets fired here, is going to go to full amplitude at 35 millivolts. That same action potential down here is going to be 35 millivolts too. Graded potentials will not do that. They can be 35 here and be 10 here. And they're irreversible action potentials. Once they start, you can't neutralize them. Once they start, it's always going to go to completion. There's a refractory period. There's two of them. There's an absolute and a relative refractory period. During an action potential, for a few milliseconds after it starts and goes through, it's very difficult or impossible to stimulate that region of the neuron to initiate another action potential. You can't initiate another action potential during the depolarization and repolarization. So until the sodium gates close completely, they can't be reopened. So unless they're completely closed, you can't reopen them. That's the absolute refractory period. Once they're closed, a new action potential can be triggered, but only with an exceptionally strong stimulus because we're hyperpolarized. Meaning it has to even be even a stronger, more excitatory signal than it was when it was resting membrane potential, because we're now at -72 instead of -70. That's the relative refractory period, and that's why that hyperpolarization is so important. Is that the relative refractory period will last until the potassium channels completely close. And a new stimulus can reopen the sodium channels to depolarize the membrane, but potassium is still diffusing out. So it has to be a very strong signal. Has to be a very strong local graded potential. Questions? So absolute is because of the sodium gates. You can't reopen them until they're completely closed. The relative refractory period is because of the potassium channels, because they're still open, still making the inside of the cell more negative, making it harder for it to reach threshold. So comparison of local versus action potentials. We've kind of seen this before, but not summarized. Local potentials are produced by gated channels. So these are ligand gated channels that are opened a lot of times by neurotransmitters binding to the channel. Action potentials are produced by voltage gated channels. Local potentials may be a positive or negative voltage change. So they can be depolarizing or hyperpolarizing. Action potentials are always depolarizing. Local potentials are graded proportional to the stimulus strength. Action potentials are the same every time, regardless of what the initial strength stimulus was. If it reaches threshold, it's going to go to maximum every time. Local potentials are reversible. As soon as the stimulus ceases, the potential goes away. Action potentials, once they're started, are irreversible. They're going to go to completion every time. Local potentials only travel for a short distance from the point of origin. Action potentials are self-propagating. This is going to be important later too. Action potentials are self-propagating. So one action potential will initiate the next action potential, all the way down the axon. Local potentials are decremental, meaning the signal is going to grow weaker with distance away from where it started. Action potentials are non-decremental. The signal maintains the same strength regardless of the distance. So it has the same strength as it did when it started in the trigger zone, as it has at the end of the axon, next to the axon terminus. So, question, maintenance of the resting membrane potential is possible due to what?
D.
It is D, correct. So if you remember, we had non-myelinated axons and we had myelinated axons. Myelinated axons, excuse me, let me start with non-myelinated axons. So non-myelinated axons or unmyelinated axons have voltage gated channels completely along the whole entire axon length. Myelinated channels, myelinated axons, on the other hand, are scarce, except for the nodes of Ranvier. Remember, those are the gaps where we don't have myelin. In the places where we have myelin, there's fewer than 25 channels per micrometer squared. In the nodes of Ranvier, there's 2,000 to 12,000 channels per node, per micrometer squared. Remember that for right now, remember that action potentials are self-propagating. Okay, this will make a lot more sense. Say it again.
I have a quick question. I wasn't sure if you were able to maybe like is there a certain stimulus or example you can give that would cause an action potential versus and what would be like a graded potential?
Can you wait on that one? Hold off on it, because yes,
Yeah.
there is. Yes, we'll see it. So remember they're self-propagating. So when you're looking at an unmyelinated action potential down a nerve fiber, the action potential starts at the trigger zone. Starts at the trigger zone. And sodium diffuses for a short distance along the plasma membrane. Starts moving down the plasma membrane. The resulting depolarization, remember what we saw before, it's a positive feedback loop. So that when sodium starts to enter, it changes the voltage just a little further down the membrane, causing more sodium channels to open. So the depolarization of those first channels, the opening of those first sodium channels causes depolarization of the membrane just further down, a little further down, causing an action potential there. And the potassium and sodium channels are going to open just as they did in the trigger zone, and a new action potential is going to be produced. This is a self-propagating action potential all the way down the axon. It just keeps going all the way down. Why does it not move? Why does it go one direction? It doesn't go backwards. Why does it not go backwards? Ions, they're going to move in both directions. Once they move into the axon, they're going to move in both directions. Why doesn't the action potential keep going? You know, why doesn't it go both directions? Because of the absolute refractory period. You can't reopen sodium channels until they're completely closed. That's the reason it keeps going in one direction. And so the steps 1 through 3, these steps of sodium diffusing in, causing more sodium channels to open, and then potassium channels to open, is going to occur all the way until the end of the axon. So it's just continually going like this all the way down. Questions?
Can I just clarify what you just said, if you don't mind?
Go for it.
So you're saying that the ions, which is the sodiums and the potassiums, will go both directions, but the axons only go one direction.
The refractory period is going to prevent it from being able to go both directions. You can't, the reason the action potential only moves one direction is because of those refractory periods that prevent it from going backwards.
Okay.
Does that help, Zena?
Yeah. Yes.
So the result of this continual propagation of this action potential all the way down causes this non-interrupted wave called continuous conduction. That's what we get in an unmyelinated axon, is continuous conduction all the way down. and I kind of skipped ahead a little bit here, but this this continuous conduction is this repeated nerve signal, repeated action potentials that keep getting initiated all the way down. Are these traveling waves of excitation produced by self-propagating action potentials. Only the membrane ahead is sensitive to the stimulation because of those refractory periods, preventing the nerve signals from moving backwards. Justin, tell me this is making a lot more sense than it did the other day.
It's definitely making a lot more sense.
Awesome. Okay. So this continuous self-propagation works like the wave-like amplitude. You've seen this in like big crowds, where it moves all the way from the beginning to the end and doesn't lose amplitude. So how come, and this is what's going to be important, is how come myelin makes things faster? It still moves down non-unmyelinated axons. It just doesn't move as fast. Voltage-gated channels are scarce in myelinated coded regions. In these regions where there's myelin, there's not a lot of channels. Also means there's not a lot of leaks. When you put a lot of, if you've ever been shocked by an electrical cord, you don't forget it. But there's not a lot of leaks. If there's not a lot of channels there, there's not going to be a lot of leaks. So voltage ion channel is scarce in the myelin-coated regions in between the nodes, the internodal regions. So no action potentials occur in these regions. When sodium, but remember there's a lot of channels in the nodes. Remember there's 2,000 to 12,000 per micrometer squared. There's a lot. So when the sodium enters, it starts to diffuse down. Remember all the sodium ions, they're all positively charged, right? And do same charges repel one another or do they attract?
Attract.
Repel.
Oh, wait. I was funny. What do we think? Are they attracting or repelling?
I would say repel because they're the same, but opposites attract, right?
Right. They're repelling one another. So as they move in, and there's no leaking, once they move in, they're stuck in this thin little rod. I'm gonna scoot in just a little bit more. Bear with me here. I like that I can see all you guys' faces. I appreciate it. So they're moving in, and they don't like to be that close to one another. They want their personal space. So as they move in, they immediately start repelling one another and moving down. But there's no way out of this until you get to the next node because there's very few channels. And so they all start moving in and can only get out when they reach the next node. Hi, Albers. You guys hear my cat squawking at me? She's very demanding. You gonna show your face? Come here. She says, you have a cat on your dog on your lap. I don't want to show my face. This is my Abby. She says, Mary, leave me alone. She's the one who really likes her personal space when it comes to the other cats and the dogs. So each sodium, as it moves in, repels all the other sodium ions. So no one sodium ion is going to move very far in this region, but their electrical repulsion is going to result in them an energy transfer down the membrane. This is kind of like a crowded subway car. The doors open. You've ever ridden a subway like in Washington, D.C.? I honestly have never been down to Philadelphia or Pittsburgh, so I don't know if they have subways there or not. But you get into a subway car. Everybody is trying to get some personal space. And people keep moving in. So everyone has to push to get to the rear of the car to make more room. No passenger is actually going to move from the door to the rear, but the crowding and transfer from person to person forces those at the rear to move just a little further down. So the events at one node are going to create excitation at the next node. The major reason why this myelination is so much or this conduction is so much faster is rather than having to do this gradual reoccurring all the way down. And even though it's fast, when we have, actually, let me draw it, bear with me. Using my finger doesn't work very well sometimes. So here's my membrane. Here's my unmyelinated axon. The action potential continually goes like this all the way down. Constantly moving, pretend they're all the same amplitude. All the way down. However, oh, sorry, my cat bumped my arm. Just, you're not helping. So because most of the area is covered with myelin, rather than having to continually move it down and have 3 or 4 action potentials within this region have to be reinitiated. It only has to be initiated here, then it travels all the way down and gets initiated here, travels all the way down, gets initiated here. Sorry, my cat bounced my arm again, but you get the idea. She's very demanding when she wants to be. Huh.
So it's almost like one is like a car in traffic and the other is like being on like a bullet train and only stopping at stops, right? Like
right.
longer
right.
spaces.
Right, exactly. That's exactly it. So the myelin is going to assist conduction in two ways. It's going to seal up leaks, prevent leakage of sodium ions out of the axon. And the result is that there's a higher concentration of sodium in the membrane inside the axon, which allows for energy transfer to move much faster. There's also a greater separation of charge between the intracellular fluid and the extracellular fluid. So if you think about this, here's the positive charges, negative charges out here. This membrane is not very thick. And if you remember from before, this charge, this potential is only on the surface of the membrane, only on the membrane. It's not throughout the axon. So we've got these positive charges here, we've got negative charges out here, and those negative charges out here can keep these positive charges in place by keeping an attraction to them. Well, when you wrap up a bunch of myelin and insulate these positive charges from the charges out here, they're going to spread a lot further along. If you think of it, and I'm not against relationships, but if you think about it, if you're not stuck with somebody, you can go wherever you want. But once you're attracted to someone and you make a commitment, you're stuck. And living really far away from them is not going to work. So think of it the same way here. So the cations and the anions in the intracellular fluid and extracellular fluid are less attracted to each other. So the sodium channels move much freer within the axon when it's myelinated. Questions? Okay. The rest of this chapter is going to go a little faster, I hope, but I think it's important to understand that. So conduction in the myelinated nerve fibers is really fast in the internodal regions. Conduction is slower at the nodes of Ranvier, but you only have, that's the only place where you have to initiate an action potential again. That's the reason it's so fast. So conduction occurs fast in the internodal regions with excitation at one node of Ranvier creating excitation at the next node. On a summary of this, I personally, I think Justin's was the best idea, was the bullet train versus one that stops everywhere. But another way, another analogy of this is dribbling a ball versus bouncing it. And actually, that's not even right. I can't believe I just said that. Dribbling a ball versus throwing it to somebody. Speed is determined by myelination, temperature, and diameter of the axon. Temperature, if you increase the speed, if you increase the heat, increase the energy, which means you're going to increase the heat, you give ions a lot more energy than they had before, and they're going to move faster. Any questions about this? So nerve conduction, nerve firing and conduction of an action potential. Bear with me just for a second here. Zoom out just a little bit. You receive the inputs at the dendrites or the soma. The action potential is triggered at the trigger zone. It's initiated at the trigger zone, which is at the axon hillock. Then it's conducted down the axon to the axon terminals. And the axon terminals are the output zone, where transmitter is going to be, these are going to synapse with the dendrites and soma of other neurons. So when we're looking at synapses, we're looking at a synaptic relationship between neurons. Signals arrive via presynaptic neurons. And these presynaptic neurons release a neurotransmitter at the synapse that is going to diffuse over and bind to a postsynaptic neuron that's going to respond to that neurotransmitter. So the presynaptic neurons can synapse with a dendrite, a soma, or an axon of a postsynaptic neuron. Remember, synapses are, I think of them as like snaps on shirts. It's a coming together of the two different things. The more synapses a neuron has, the more input it can receive. So a motor neuron, spinal motor neuron, remember that comes out the anterior horn of the spinal cord, is covered with about 10,000 axon terminals. All these little spots right here, synapses. With 8,000 of those synapses ending on its dendrites and 2,000 on the soma. In the cerebellum, where we make a lot of decisions about motor movement, one neuron can have as many as 100,000 synapses with other neurons. Two different types of synapses. We have electrical synapses, which are where adjacent cells are joined by gap junctions, and ions diffuse directly from one cell into the next. There's synchronous activity. Remember what gap junctions are. They're tunnels in between cells. So ions can diffuse directly from the presynaptic neuron into the next cell. So these are found in some neurons, neuroglia, and cardiac and unitary smooth muscle. Chemical synapses are where neurons communicate via the neurotransmitters. These are sites of memory and learning, and targets of many prescription drugs, and sites of action for many addictive drugs. This is why drugs are, some things are addictive, like caffeine. Which most of us can say we have an issue with. Electrical synapses, an action potential is going to spread from one neuron to the next via gap junctions. There's no transmitter needed. They're extremely fast, and they allow for two-way conduction. This provides for coordinated contractions of all muscle fibers. So sometimes it's not a neuron that's the postsynaptic cell. It can be a muscle. It can be a gland. But it provides for coordinated contractions of all muscle fibers all at once within cardiac and visceral smooth muscle, and limited eye regions in the brain and eyes, because of the presence of those darn gap junctions. A chemical synapse is a little different. It doesn't have those gap junctions. The axon terminal contains synaptic vesicles. And in between the presynaptic neuron and the postsynaptic cell, there's a synaptic cleft that's 20 to 40 nanometers. So 1 to the negative 9th of a meter. So wait, let me think here. 1 billionth of a meter is 1 nanometer. So there's a small synaptic cleft, and on the postsynaptic neuron or cell, there's a bunch of receptors for the neurotransmitter. In all synapses, there's a synaptic delay. Guys, remember when I asked you at the end of the last lab, which one was faster, monosynaptic reflexes or polysynaptic reflexes? The reason monosynaptic reflexes are so much faster is because of this synaptic delay. When they're monosynaptic, you only have one. When they're polysynaptic, you have more than one delay. So in all synapses, there's this delay. This is this is defined as the time from the arrival of the signal at the axon terminal to the beginning of the action potential of the postsynaptic cell. So this is the time where that signal arrives here. The synaptic vesicles are released into the synaptic cleft. The neurotransmitter gets released. It diffuses over and hits the receptor. That's the time of the synaptic delay. Any questions about this? Neurotransmitters are molecules that are synthesized by a neuron, released when a nerve signal reaches the axon terminal, and has specific effects on the target cell's physiology, the postsynaptic cell, which is the target cell. Most neurons are released via exocytosis. More than 100 neurotransmitters have been identified, but most fall into 4 major categories. Acetylcholine, amino acids. You remember what amino acids are, right? Maybe. Amino acids are the subunits that make up proteins, or the monomers that make up proteins. So we have some amino acids that act as neurotransmitters. we have one, GABA, which is a derivative of, what is it? It's kind of a derivative of lysine, which is an amino acid. Another category are monoamines that are synthesized by amino acids by removing the carboxylic acid. These include catecholamines, epinephrine and norepinephrine, and dopamine, serotonin, and histamine. Many of these you've heard of before. You've heard of people who carry around an EpiPen because epinephrine will stop an anaphylactic reaction. The antidepressant Prozac works on slowing down breakdown of serotonin. A lot of our Parkinson's drugs work on maintaining dopamine levels. Antihistamines. The fourth category is adenosine and adenosine diphosphate. You know that adenosine is one of the nucleotides. Adenosine triphosphate is an energy currency. Well, it's also a neurotransmitter. Some other categories of neurotransmitters, gases, nitric oxide and carbon monoxide. We always think of carbon monoxide as being a toxin to you. But in some cases, it's a neurotransmitter. Specifically, I can tell you we know the most about nitric oxide. It's synthesized as needed. It's not stored in synaptic vesicles, so it's not released via exocytosis. You remember from up here, it says most are released via exocytosis. Nitric oxide is not. It will diffuse out of the axon terminal and diffuse into the postsynaptic neuron rather than bind to a receptor. It's also a neuromodulator, which we'll talk about later. There's also neuropeptides, which are chains of 2 to 40 amino acids. They're stored in secretory vesicles, and they can also function as neuromodulators. Questions that I may or may not be able to answer because there's a lot of neurotransmitters. Neurotransmitters can be excitatory or inhibitory. Their effect depends on what type of receptor is found on the postsynaptic neuron. The receptor is going to determine what effect that neurotransmitter has. So some neurotransmitters are both excitatory and inhibitory, meaning they're more likely to help that neuron reach an action potential, the postsynaptic neuron, or they can also cause it to not have an action potential triggered, depending on what the receptor does. So the receptor is going to govern what effect the neurotransmitter has. Acetylcholine is a common neurotransmitter that's released by many peripheral nervous system neurons. And it's excitatory at the neuromuscular junction when the postsynaptic cell is a muscle, but it's inhibitory at all other synapses. So there's multiple receptor types for each type of neurotransmitter. Now we're going to talk about how does this synapse action potential trigger occur? How is the neurotransmitter released? The neuroaction potential comes down this presynaptic neuron. And when it gets to the axon terminal, to this terminal bouton, it's going to open voltage gated calcium channels. As we move forward, you're going to learn that calcium is really good at triggering exocytosis and triggering things to happen within a cell. So the arrival of the nerve signal, that action potential at the axon terminal opens voltage gated channels. The calcium enters the terminal and triggers exocytosis. It binds to those vesicles, those synaptic vesicles, and causes them to fuse with the plasma membrane here and release the neurotransmitter. So the vesicles release their neurotransmitters, and the empty vesicles recycle back into the cytoplasm to be refilled by acetylcholine. This is what we're seeing right now are the steps of an excitatory cholinergic, because it's acetylcholine synapses. Acetylcholine diffuses, once it's released into the cleft, diffuses across the cleft and binds to ligand-gated channels on the postsynaptic neuron or postsynaptic cell. And the result is that sodium enters the postsynaptic neuron. Remember what sodium does when it enters a cell, it depolarizes that membrane, producing a local postsynaptic potential. And each postsynaptic potential, if it's strong enough or persistent enough, will open up voltage-gated channels in the axon hillock and induce an action potential. Questions? During the events involved in information transfer across a chemical synapse, which of the following steps would be directly interrupted by exposing a neuron to a calcium channel blocker?
Recording in
What do you think? If we block calcium from coming into the presynaptic neuron, what happens?
Is it D?
D? It's not D.
B.
See? Go up a little further. Where does calcium enters?
It would be A, right?
It's A. Because if we block calcium from entering, we're not you're not going to get exocytosis of the neurotransmitter. Drugs are really good at modifying the effects of neurotransmitters. You can stimulate or inhibit the synthesis of a neurotransmitter. They can block or enhance the release of neurotransmitter. They can activate or block specific receptors that bind neurotransmitter. They can stimulate or inhibit the removal of neurotransmitter from the synaptic cleft. Nicotine works by binding to receptors that normally bind to acetylcholine. When acetylcholine binds to these axon terminals receptors, that causes the release of dopamine. And the increased feeling of dopamine entering the cell is what causes nicotine to be so addictive. You get used to that feeling of having extra dopamine around. Because nicotine acts like acetylcholine. It's amazing when you start looking at different drugs and things that are very addictive like nicotine, which if you know somebody who smokes or you do smoke, you know how potent it is as an addiction. This is why. One of my best friends, one of my partners in crime when it comes to anatomy and physiology. We don't talk very often, but we talked this weekend for the first time in like 6 months. She knows everything about nicotine. She knows everything about it. She knows, she told her kids when they were little that if they ever decided to start smoking, their heads would turn blue and pop off. And shockingly, both of her kids have never taken up smoking. But she started smoking when she was 13. She has never been able to quit. And she's now probably, I'm trying to think, I think she's 46, somewhere in there. It's nasty. This is why. It's next to heroin in its ability to form addictions, potency-wise. So in opposition, synapses that employ GABA work by the same mechanism as acetylcholine. However, the GABA receptor on the postsynaptic cell is a chloride-gated channel. If chlorine, which is negatively charged, enters a cell, what's going to happen to the membrane potential? You guys, if chlorine, which is negatively charged, enters a cell,
It'll become more negative.
It becomes more negative than it was before. I don't see Sam's face now because I only see like 6 faces at a time. But Sam, this is your answer to things that are excitatory versus inhibitory. Things that easily allow sodium in are going to be excitatory. Things that easily allow chlorine in are going to be inhibitory. Hi, Ivy. She's a cute kid. She's a very cute little girl. And I still love the name.
you. I'm taking notes.
Of course.
I'm taking notes.
So Sam, chlorine and potassium channels, things that easily open chlorine and potassium channels are going to be inhibitory because they're going to cause the inside of the cell to become more negative than it was before. Channels that allow sodium in are going to end up being more excitatory. An adrenergic, meaning it's neurotransmitters like epinephrine and norepinephrine or other monoamines are going to act via second messenger systems. You remember this when we talked about cell biology before, acting through amplification using cyclic AMP. The receptor, rather than being an ion channel, is going to be a G-coupled protein receptor. So you activate the G protein, the receptor binds to the G protein. It activates and it creates cyclic AMP, which then goes on to do multiple things, which may include opening sodium channels. So an unstimulated norepinephrine norepinephrine receptor is coupled to a G protein. The G protein binds to the norepinephrine, binds to the G protein. The G protein gets activated and activates the adenylate cyclase. You guys, I have to stop just for 2 seconds. Ivy, I ordered the cow eyeballs today. I can tell you that normally in this class, I don't do that. But then when you said you were so excited about the eye, I thought we got to dissect the cow eyeball. So I got some cow eyeballs and they're coming in the mail.
I told her after our last class, and she started studying her anatomy of the eye so that she recognizes structures when we do the dissection.
Okay, I'm going to quiz you. I'm going to quiz you come in 2 weeks.
Okay, I'll try.
Maybe I don't want to get into this. I understand.
Or tonight.
Okay. So, G proteins get activated. They activate adenylate cyclase. Adenylate cyclase creates cyclic AMP. Cyclic AMP goes on to have multiple effects, including activating other enzymes, activating gene transcription, and opening sodium channels. Guys, if you have questions, please just interrupt me and stop me. Although these channels, these adrenergic site amplification versus synapses are slower, they work by signal amplification. So one cyclic AMP can go on to do multiple things. One norepinephrine neurotransmitter can activate multiple cyclic AMPs. And one cyclic AMP can go on to activate multiple things. So you start by activating one receptor, which then activates multiple enzymes that create cyclic AMP. Then each cyclic AMP goes on to activate a bunch of other things. So rather than just opening one ion channel per neurotransmitter, now we're one thing is activating a lot of other things.
So now I'm just wondering, what would open a chlorine channel? Like, is there a certain drug that would open it up?
Chlorine channel?
Yeah, like,
GABA
would it be
would.
like a certain drug, or would it be something like certain
It could.
food?
It
Or
depends. It can be a lot of things. There's a reason that some drugs are relaxants and they slow you down. Opioids slow you down by slowing down nerve impulses. And they do that by working on stopping the triggering of action potentials by hyperpolarizing the membrane. Does that make sense, Sam?
yeah, thank you.
Elimination of so we like neurotransmitters, we love neurotransmitters, but we don't want them around all the time. We want to be able to get rid of them. We want to get an action potential trigger, but then we need it to go away when we want it to go away. How do we get rid of it? There's 3 different ways. It can get degraded, where enzymes in the synaptic cleft get broken down. They break down the neurotransmitter. Like acetylcholine esterase breaks down acetylcholine. It gets retaken up. So the neurotransmitter gets retaken up into the presynaptic neuron.
It's time to go to bed.
What?
Sorry, I was talking to my kids. I was telling them to go to bed.
That's funny. Okay.
Yes, it is time to go to bed.
Don't say that. Don't say that, Lauren. You're gonna. We're on a kick here. I want to finish this tonight. Well, don't say that. The other thing is the neurotransmitter can diffuse away from the synaptic cleft. So it either gets degraded, broken down, it gets taken up by the presynaptic neuron, or it just diffuses away. Prozac works really great because it blocks the reuptake of serotonin. So it stops the signal from being removed as fast. The neurotransmitter stays in the cleft longer than it would normally. It blocks it from being retaken up. Any questions? Neuromodulators. So we have molecules that are secreted by neurons with long-term effects on entire groups of neurons rather than brief, quick effects of neurotransmitters. They function to increase the release of neurotransmitters by presynaptic neurons. They can adjust the sensitivity of a postsynaptic neuron to a neurotransmitter in the cleft. Or they can alter the rate of neurotransmitter reuptake or breakdown. So they alter if that neurotransmitter is actually released into the synaptic cleft. They can alter how the postsynaptic neuron is going to respond to the neurotransmitter. And they can alter how long that neurotransmitter sticks around in the cleft. I honestly think this is probably the neatest part of this chapter, because we all know people who struggle, who are on an antidepressant or struggle with an addiction. This is what those things work on. So examples of neuromodulators: nitric oxide diffuses into the synaptic postsynaptic cell and activates second messenger systems like the cyclic AMP. Relaxing the smooth muscle, allowing the dilation of small arteries and increasing blood flow. It's the reason Viagra works the way it does. Neuropeptides such as enkephalins or endorphins inhibit spinal neurons from transmitting pain to signals to the brain. This is why when you're working out and you cut your foot, you may not recognize that you've cut your foot until you stop working out, as I learned from experience, because your endorphins are going so high. I had an experience. I was out. I used to run. I don't run now because I have a curve in my lower spine, so I quit running. But I still like to work out. And so I cycle a lot. But at one point in my life, I used to run a lot. And I was up in the northern part of Minnesota, running around Itasca State Park, which is really kind of neat because it's right on the Canada border. And there's lots of trails up there. And so I was running around on this trail, paved, nice trail, and got back to my cabin and looked down at my foot and realized that my foot was soaked in blood. Because while I'd been out running, one of my toenails had cut my other toe. And so when I finally quit running, I looked down and realized that it had been bleeding for a major part of my run. And I didn't feel it until I got back because my endorphins were so high. Other modulators are hormones such as dopamine, serotonin, and histamine. So the word neurotransmitter, hormone, or neuromodulator is not defined by the chemical itself. Any chemical can be a neurotransmitter, or hormone, or neuromodulator. It depends on the role that whatever that chemical plays in that context. So a given chemical can be all three, depending on what receptor and what context it plays, what's its function in that moment. That's why histamine works as an anti-allergic, allergy hormone. However, it's also a neurotransmitter. ATP is a really great energy currency. It also is a neurotransmitter. Neural integration. So the more synapses a neuron has with other neurons, the greater its information processing capabilities. A pyramid cell on the cerebral cortex has about 40,000 synaptic contacts with other neurons. Remember that one neuron in the cerebellum can have up to 100,000 synapses. Because the more information you have, the better your processing abilities. The more abilities you have to receive information, the more information you can process and take into account. That's why knowledge is power. The more you have, the better off you are. So neural integration is a neuron's ability to process information, store and recall it, and make decisions. It's based on postsynaptic potentials initiated at the axon's trigger zone. So how many excitatory signals do you get? How many inhibitory signals do you get? Is going to decide what the next result is. So we have excitatory postsynaptic potentials. These are the ones that make the membrane more depolarized, to make it closer to threshold. So they make a neuron more likely to initiate an action potential. Sodium flows into the cell, making the inside of the cell less negative, meaning it's closer to the threshold. It's an excitatory postsynaptic. An inhibitory is going to hyperpolarize the signal, the membrane, making it more negative than the resting membrane potential. Can either cause chlorine to flow in or potassium to flow in, or potassium to flow out, causing the inside of the cell to become more negative, pushing it further away from threshold. So one given excitatory postsynaptic potential or one inhibitory postsynaptic potential will not dictate whether or not a neuron will fire an action potential. It just changes the rate of firing by stimulating or inhibiting the production of more action potentials. This is going to determine the probability of getting one to occur. Excitatory is going to increase the probability. Inhibitory is going to decrease the probability. So if potassium leaves the cell, it's going to result in an inhibitory postsynaptic potential. If chlorine enters the cell, it's going to result in a more negative membrane potential, meaning an inhibitory postsynaptic potential. If sodium enters the cell, it's going to result in an excitatory postsynaptic potential because the membrane potential is going to become more positive closer to the threshold. So normally a single excitatory postsynaptic potential will not initiate an action potential. It's got to summate, meaning multiple postsynaptic potentials are going to summate, and the neuron is going to respond to their net effect. And this response occurs in the trigger zone where we have the most amount of voltage gated channels. Questions? Guys, I want to keep going because I want to get into the spinal cord. If we can, I'd prefer to get into the spinal cord, and we're coming to the end of this. Two modes of summation. So two different ways that things can sum. Remember that these excitatory and inhibitory postsynaptic potentials are, bear with me. they're decremental. Meaning if the stimulus stops, they stop occurring and it goes back to normal. If the further away you get from where they started along the cell body of the soma, the weaker the signal becomes. So we have two things here. We have temporal summation, where one presynaptic neuron generates so many excitatory postsynaptic potentials that each potential is generated before the last one fades. And all of the excitatory postsynaptic potentials are going to add up to a voltage reaching threshold. Or we get a spatial summation where all of the excitatory postsynaptic potentials are occurring very close to one another on the cell body of the presynaptic cell or the postsynaptic neuron, which is resulting in the voltage reaching threshold. So I have my kindergarten axon done here. All my dendrites. And I have one axon, one presynaptic neuron that keeps firing excitatory signals. Pow, 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 some more. And they keep coming in and they're coming in one after another. And so the negativity of one, the negative effect that it has on the membrane, doesn't go away before the next one comes in. So pow, 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 it just keeps coming. And the result is that you get that it sums to get where you reach threshold and you fire an action potential. That's temporal summation, where they keep coming in so fast that they're not, the resting membrane potential doesn't have time to return back to the normal. And that's what allows you to reach threshold. Or another one is that I have multiple ones coming in. And this one fires an excitatory one. This fires an excitatory one. This one that's right here fires an excitatory one. And they do it all close to one another. And this close within time, they're all close to one another. And the result is that between all of their negative effects, you reach threshold. All of their excitatory effects, you're going to reach threshold. That's spatial summation. Any questions? Sam, I think your Yoda has a question. Your baby Yoda in the background has a question. Says Mary, I don't know what you're talking about. Sam, do you see the baby Yoda? I see the baby Yoda. It looks like,
Oh, this one?
yes,
Oh, yeah.
just helping. Okay. She's like, I don't know what she's talking about. Okay. It looks like a baby Yoda to me. All right, so coming back. So all of the one presynaptic potential neuron keeps firing so many excitatory postsynaptic potentials that they end up reaching threshold at the postsynaptic neuron. Spatial summation is when they're coming in from multiple presynaptic neurons. And all of them summing together adds up to reaching threshold. Suppose the excitatory postsynaptic potential summate in a presynaptic neuron in response to simultaneous stimulation by neurotransmitters, glutamate, serotonin, and acetylcholine, released by 3 separate presynaptic neurons. Is this an example of spatial or temporal summation?
Spatial.
It is, it's spatial. Because and why is that the case, Mr. Justin?
Because it's multiple neurons and
Right.
multiple sources.
Yes, exactly. Good job. Which of the following is true about a true statement about neurons? Signals can travel in either directions in both dendrites and axons. Dendrites conduct signals towards the cell body. Axons conduct signals away from the cell body. Dendrites conduct signals away from the cell body, and axons conduct signals towards the cell body. Signals can travel in either directions in the dendrites, but only away from the cell body and axons. Or signals can travel in either direction in axons, but only toward the cell body and dendrites.
Is it B?
B is in Bravo, Sam? Yes. Why do you say that?
Because it makes the most sense. Because earlier you said that, like, the signals can only travel in one direction, and the dendrites are located closer to soma, so it has to go through the soma down the axon.
That's perfect. Yes, that's right. Membrane depolarization is caused by what?
E echo.
E echo? Is that depolarization? Look at it again. Who was it who said that? Of course, they're not saying it now. All right, someone else. What do you think? It's A. So sodium ions are moving into the cell. Which of the following is not true about synapses? Somebody tell me, what do you think?
I'm gonna guess C.
You're good. Yes, it's C, because they don't bind to both sides. It's only binding to one side, right? But sodium potassium pumps help maintain the membrane. Louis, help maintain membrane polarization by what?
Is it D?
B is in Bravo, Marissa?
D is in dog.
D is in dog. You're close. Does it pump sodium into the cell? Or does it pump sodium out?
From sodium in and potassium chloride out.
So wait, say it again, Zena.
I was saying it pumps sodium in and then potassium out.
It's going to do the reverse. I think that's what Marissa was going to say. Is that right? Okay. It's
Yeah,
going to do the reverse. So it's pumping sodium out while pumping potassium in.
So does the chlorine go into? No, I have it mixed up.
Chlorine doesn't normally play a part in that. It will actually follow sodium, but it's not involved with that pump.
Okay, so it just follows the sodium.
It does. It does.
No, you didn't like that.
But for the most part, don't worry about it.
Okay.
Presynaptic facilitation. One presynaptic neuron can enhance the effects of another presynaptic neuron. When one neuron releases a neurotransmitter that allows for calcium channels on another presynaptic neuron to stay open longer, causing neurotransmitter to be released for longer. Remember what calcium does? It causes the release of the neurotransmitter. So neuron F, the facilitating neuron, accentuates the transmission of neuron S. Also have presynaptic inhibition, where one presynaptic neuron inhibits the effects of another presynaptic neuron. So it's used to reduce or halt unwanted synaptic transmission. You've seen this before. I know Zena has small kids. Justin has small kids. Sam has small kids. Your kids learn this really quick. When you bang your arm or you bang your knee, what's the first thing you do? You rub it, right? Because what it does is it inhibits pain signals. If you want to give a really good immunization where people don't feel it, Massage the skin for a second before you stick the needle in. I can tell you that getting all my little COVID boosters, who knows how to do that? I can tell you who knows how to do that and who doesn't know how to do that, depending on how much I felt. So presynaptic neurons inhibit iRuby. Presynaptic neurons inhibit the effects of other presynaptic neurons. Used to reduce or halt unwanted transmission. Neuron I releases an inhibitory transmitter, that GABA, that prevents calcium channels from opening on another presynaptic neuron. And the result is that neuron S, which is the postsynaptic neuron, releases less or no transmitter. Neural coding. You guys, do you have questions? Just keep going here. If you have questions, please interrupt me. Neural coding, the mode by which information about or around the body is communicated in meaningful patterns of action potentials. The theory is that there's a labeled line code. The basis of this is that each nerve fiber leads from one receptor that recognizes only one stimulus. Any stimulus received from a nerve is interpreted by the optic nerve, is interpreted as a form of light, because the receptors in the eye only see light as a signal. You've done this before, and I'm glad I'm not wearing makeup. I don't wear makeup when I teach online, so you see what I really look like. But right now, we're seeing, you close your eyes and rub your eyes. What do you see when you do that? Someone do it. Maybe not, preferably not someone wearing makeup. What happens when you do that?
You see floaters.
You see floaters? Are you sure? Are they floaters? What do they really look like? What do you really see?
Like dark spots of light, or dark shadowy.
They're kind of like little splashes, aren't they? Little tiny ticking things. Because what that receptor is, the nerve receptors in your eye are trying to recognize that pressure as a form of light. And it can't. You put pressure on your eyeball and you don't, and it's trying to put that into a form of light. That's right. That is the coolest thing that I think is in your whole entire textbook. Is that you can't, that there's no real way to recognize that. And therefore your optic nerve is trying to put that pressure and into a signal of light.
My favorite thing about light, and Marissa knows this too from working in the industry we were in for 20 years, no one's even sure that what we interpret as color is seen the same way by everyone. And it's assumed that most people see it in similar ways, but that there are people that are so, that the brain interprets that signal so differently that your red could look like my green, your blue could look like my bright purple, your, like, that it's not at all the same for anyone.
I've heard that before.
Yeah.
That's very
to piggyback off what Justin said, it kind of reminds me of a long time ago, there was this like Facebook post that was like, what color is the dress? And some people seen it as yellow and some would see it as blue or something.
Yeah. Yeah.
That dress was blue and black. I'm telling you.
Justin knows. He knows. Because Ivy told him.
Well, Ivy sees it as white and gold.
Yeah, it's white and gold. I don't know what he's
tell him.
talking about.
Justin,
It's white and gold.
Justin, you're wrong.
I know, but it's the way that our eyes perceive color. It's crazy.
It's black and blue.
We'll see.
Oh, come on.
But working in color theory, it's all about the way that our eyes perceive light is the way that we perceive color. And everyone's eye is very, very different. And some people can see a myriad of colors where some people can only see small variations in tone, and some people can only see very large variations in tone. So we're always taught that like the best colorists, the best color specialists in makeup, in hair color, we tend to see a full spectrum of color with subtle variation where a lot of people don't.
Right.
Yeah.
I'm also studying on the floor with the eye. So
She is. She's
yeah.
studying.
Awesome.
Right?
Awesome. Okay. So stimulus strength is interpreted by the frequency of how often is the signal coming in and how many neurons are firing at that time. So different neurons have different thresholds of excitation. A weak stimulus is going to excite neurons with the lower thresholds. A stronger stimulus is going to excite neurons with less sensitive, higher thresholds. Additional neurons can be recruited if a stimulus becomes stronger because the weaker ones are triggered first. And intensity is by how often are you getting part of the fire action potentials coming in. Bear with me, I want to see how far we've got 2 slides. Okay. So neurons, neural pools. Neurons function in larger ensembles called neural pools. Each pool can consist of thousands of inner neurons concerned with one particular function. They can form neural circuits, which is a network of neurons that process specific stimuli. We have diverging circuits where one nerve fiber branches off and synapses with several postsynaptic cells. And each of those postsynaptic neurons then synapses with several more. This allows for one motor neuron of the brain to affect thousands of nerve muscle fibers. We have converging circuits where input from multiple nerve fibers is funneled onto one neuron, allowing input from your eyes, your inner ears, and stretch receptors in your neck to be directed into a sense of balance. We have reverberating circuits where neurons stimulate each other in a linear sequence. But some neurons later in the path send axon collaterals back to reinitiate the stimulus. These kinds of circuits send repetitious signals to your diaphragm and intercostal muscles to make you inhale. This is what opioids actually work on too. to slow down breathing. When the circuit stops firing, you exhale. The next time it fires, you inhale again. In parallel after discharge circuits, where a single presynaptic neuron stimulates a group of neurons, which then synapse with a common postsynaptic cell. Explains why you can stare at a lamp, close your eyes, and continue to see that lamp. And you guys, that's actually the end. We wanted to start the spinal cord. We won't. Obviously, we're not going to. It's 8:39. Bear with me just for a second here. I think, and if I remember right, this PowerPoint, of course, it's not going to open. This PowerPoint is significantly shorter. I want to finish it within the 3 hours on Tuesday and start the brain. So plan on it. And then we'll do the brain in lab next week and do the eye and special senses the following week. So in 2 weeks, Ivy, we're doing the eye. And you guys have a wonderful night, okay?
You too.
Thank you. You too.
Thank you. Have a great night.
You
Have a good night.
Have a good night.
too.
So I do have a question. I
Oh.
wanted to wait till like after class, like almost the class ended. How does the like epinephrine